Welcome back to the G protein videos. What I want to do in this video is I want to explore. So what we did in the last video is we looked at how the G protein system actually functions when it's activated, but we have to have a way to terminate the activity, right? So we've got all this cyclic AMP floating around. We've got all these, we got these proteins that are phosphorylated, right? So we have to have a way, and actually I should go ahead and put that this protein is also phosphorylated, right? So we have to have a way to terminate the response. And we're going to do that in three ways. The first way, which is actually sort of the easiest conceptually in my opinion, is we're going to terminate cyclic AMP activity. Well, how do we do that? Well, we're going to have water floating around, right? And there's going to be an enzyme. This enzyme specifically is called a phosphodiesterase. And a phosphodiesterase essentially is going to hydrolyze a phosphodiester bond, right? So let's actually look at the mechanism of how it works, and then it'll help you understand what it's doing. So this lone pair is going to do a nucleophilic attack on this phosphorus, right? That's going to force this, it's going to force this pi bond up. It's going to kick back down, and then this bond is going to come out and extract a proton from solution, right? So it's effectively a nucleophilic acyl substitution, and what we end up generating non-cyclic AMP, and specifically it's 5'. So the phosphate is going to be on the 5' position, and that's catalyzed by a phosphodiesterase. And so you can imagine that if this enzyme becomes activated, we're going to have less and less cyclic AMP. And so that's one of the ways in which we terminate the activity. So if there's less cyclic AMP, then there's less binding of cyclic AMP to that regulatory protein right here. And so that means there's going to be less protein kinase A functioning. And you can imagine that AMP in the non-cyclic form is not going to have the same effect that cyclic AMP does, and you'd be right. Regular 5' adenylate does not have the same activity that cyclic AMP does. Cyclic AMP is very special in its activity. Only cyclic AMP is able to bind to that regulatory protein on the anchoring protein. Okay, so without cyclic AMP, or as the levels fall, the levels of active protein kinase A drop. Okay, so now that's the first way we do it. There's also a second way that we can do it, and that's by other phosphatases. And these phosphatases, what they're going to do is they're effectively going to hydrolyze off these phosphates, right? So I have water here, and then it can ultimately hydrolyze off the phosphates that were ultimately activating these enzymes, right? So there can be another water here, right? And it can ultimately remove the phosphate, right? And again, it's just a nucleophilic acyl substitution. Of course, I didn't draw the mechanism here. I didn't draw the phosphate out. But you can imagine it's just a nucleophilic acyl substitution. And effectively what these phosphatases are going to do is they're going to remove the phosphates, and therefore what were active enzymes become inactive. So without the phosphates, these enzymes, phosphorylase B kinase and glycogen phosphorylase, these enzymes are virtually inactive without the phosphates. So that's the second way we terminate the response. There's one last way we can do it, and it's actually the most complex of all the three. And it's by, actually, let me say there's actually two other ways. I just thought of another way. There's actually another way we can do it. And the way we do it is by something called a nucleotide translocation factor. A nucleotide translocation factor. And what this does is essentially, it's basically a nucleotide translocation factor. I believe that's what it's called. But anyways, what it does is essentially helps. So remember that GTP was bound to the alpha subunit, and it was normally active, right? Well, what the nucleotide translocation factor does is it effectively substitutes the GTP for GDP. And when the GTP gets lost in exchange for GDP, the alpha subunit becomes inactive, and it moves back towards the other regulatory subunit. So the nucleotide translocation factors, what they do is they interconvert GTP and GDP, and it's a reversible reaction. So when we initially bound GTP, that was also catalyzed by the nucleotide translocation factor. And also, another name for it are nucleotide exchange factors. So let me also put that in parentheses. You may see that in your textbook. They're also called nucleotide exchange factors. But effectively, what these proteins are going to do is they they reversibly move between bound GTP and GDP. So, for instance, the alpha subunit initially contained GDP, guanosine diphosphate, and when we needed to activate the alpha subunit, we used the nucleotide exchange factor and substituted GTP for the GDP. And then it became activated, and when we need to terminate the response, we simply use the same enzyme and we remove the GTP and substitute it for a GDP. So I hope that makes a little bit of sense. And of course, that's catalyzed by a nucleotide translocation factor or nucleotide exchange factor. Okay. Now the last way we can do this is the most complex. Okay. So let me draw another membrane. So here's another membrane, right? And here I'm going to draw the adrenergic receptor, right? So here's the adrenergic receptor. Again, here's the heptahelical nature of it. So 1, 2, 3, 4, 5, 6, and then 7, right? Well, it turns out, and actually, let me do this. Let me do this. Let me actually draw the G protein. So here let me just draw it as one whole unit. And you'll see why I'm doing this in a minute. So this is just G protein generic. And essentially what the last mechanism, at least of terminating the response, is a mechanism known as beta adrenergic receptor kinase. Okay, so let me write that. It is, and let me do it in yellow. So this is beta adrenergic receptor kinase. And what this enzyme does is it phosphorylates the beta adrenergic receptor, right? So you have these phosphate groups that now stick off of the beta adrenergic receptor, and it specifically does it at things like serine residues and threonine residues. But the point is that beta adrenergic receptor kinase phosphorylates the adrenergic receptor. Now that by itself does not terminate the response, but what it does do, and I'll do this in orange, is it allows the binding of another protein, and this protein is called beta arrestin. It is called beta. Usually it's BARR. ARR is the arrestin, beta arrestin, and it specifically interacts with these phosphates. Okay, and what beta arrestin does is it forces the G protein, all of the G protein, to dissociate from the adrenergic receptor. So what you end up with is something that looks like this. So you have your membrane, right? I'll try to draw it quickly. You have your membrane, right? And you have your beta adrenergic receptor, right? And of course you have the phosphates on there, right? So here's a phosphate, and then you have the beta arrestin attached to here. So here's beta arrestin. But what it causes is it causes the G protein to dissociate. And so the G protein can no longer interact with the adrenergic receptor. So if the G protein can no longer interact with the receptor, then there can be no more response, right? There can be no more adrenergic response. Now, the beta arrestin also does one other thing, and what it essentially does is it causes the invagination of the membrane, and it forces the let me do this in green. It forces the adrenergic receptor to end up inside vesicles. So the vesicles end up being intaken into the cell, and what you end up having is these adrenergic receptors inside a vesicle, right? So they're still in the membrane of the vesicle, and essentially it's just it's an invagination of the membrane. So in the vesicle membrane, you'll still have the beta adrenergic receptors, but the beta arrestin you could think of sort of induces the invagination and the intake of the beta adrenergic receptors into the cell itself. And whenever you need them again, this particular reaction is reversible. So the adrenergic receptors can end up getting back into the membrane. So these are generally the four ways in which we can terminate the response. We can terminate with a phosphodiesterase, right? And that acted on cyclic AMP to produce AMP. And then we can phosphorylate dephosphorylate the enzymes down the cascade using phosphatases. That terminates the response. And then we can also use the nucleotide exchange factor or nucleotide translocation factor and cause the alpha subunit to rebind GDP, and that terminates the response. Or we can use beta arrestin. And let me actually be perfectly clear. This beta arrestin this beta-arrestin mechanism is often called desensitization. Desensitization, and it's called that because ultimately the receptor is getting desensitized to epinephrine. So even if epinephrine was to bind, there's no more G protein associated with it. So even if the epinephrine were to bind, you might still have the conformational change, but if there's no G protein, then there's no response because there's no alpha subunit to dissociate. So I hope this video helped you get a little bit of a sense of the termination of the adrenergic response. In the next video, what we're going to look at is we're going to look at other aspects of cyclic AMP. See you soon.